Welcome. First of all, I would like to propose you a question. Why it is important to study immunology? Once I heard a proverb saying that health is not yet all in life, but all in life without health is nothing. I guess that everyone wants to live a long and healthy life, and immune system helps us to achieve this aim because it works to support our health throughout the life, and that is why it is worth studying. As you may see from this overview image, immune system includes quite a lot of beautiful cells that protect our body from a wide range of pathogens. A large number of functional elements is necessary to deal with many different pathogens. But at the same time, such complex organization make a study of immune system a challenging aim. To make this complex task more simple, we will start from basic principles of immune system organization. This will create a framework that support further understanding of more complex topics of immunology. So, basically, all mechanisms of immune system may be grouped into two big parts. Those that represent so-called innate immune response and another representing adaptive immune response. Innate immune response has cellular components such as phagocytic cells, natural killer cells, dendritic cells. And also innate immune response mechanisms include proteins that are represented by so-called complement system, which will be discussed later in more detail. The adaptive immune response also has cellular components represented by different types of lymphocytes and special proteins called antibodies. It is important to understand that innate immune response and adaptive immune response are not isolated from each other, but cooperate together, with innate immune response being activated first in case of pathogen invasion, in order to give time for adaptive immune response to deploy its highly specific, highly efficient, but time-consuming mechanisms. Now let's speak about the innate immune response in more detail. For efficient protection of the body, immune cells have to migrate to the damaged area and detect pathogen for its further elimination. How does this happen? Let's imagine that we are looking at the healthy cells of the body tissues and pathogens damaging these cells. Being damaged, cells of the body release signal molecules collectively called damage-associated molecular patterns, or DAMPs. Destruction of cells leads to an increase in the amount of DAMPs with the highest concentration in the focus of damage. Immune cells have special receptors that are able to detect DAMPs and guide cellular migration towards the concentration gradient. When immune cells reach the focus of damage, they have to detect pathogens that should be eliminated. To understand how this happens, let's look at this illustration and imagine that we are looking at different pathogens. As you may see, they all have some typical similar structures that are normally not present on the surface of healthy cells of the body. Collectively, such structures are called pathogen-associated molecular patterns, or PAMPs. For their detection, cells of the innate immune response use special pattern recognition receptors. When cells of the innate immune response detect pathogen-associated molecular patterns with the help of their pattern recognition receptors, these cells may start to attack invaders. Pattern recognition receptors detect PAMPs that can be extracellular or intracellular. To target different types of PAMPs, innate immune cells have different types of pattern recognition receptors. For example, toll-like receptors or TLR. They are represented by plasma membrane TLRs that recognize bacterial products such as lipopolysaccharide, and also there is a group of toll-like receptors that is represented by intracellular structures or intracellular TLRs that recognize viral and bacterial RNA and DNA. These nucleic acids associated with pathogens may appear inside of the cells in case of phagocytosis of bacteria or viral infection. Another type of pattern recognition receptors are node-like intracellular receptors that recognize a wide range of compounds, including products of necrotic cells and microbial products. Yet another type of pattern recognition receptors represented by C-type lectin receptors that are able to detect fungal components. G-protein coupled receptors can recognize short bacterial peptides containing N-formylmethionine residues. RIG-like receptors can recognize RNA of the pathogens. Also, so-called DNA sensors can recognize DNA of the pathogens. So far, our attention was focused on the cellular mechanism of the innate immune response. But it also has a powerful mechanism represented by the proteins that perform their function outside of the cells. These proteins are collectively called complement system. The complement system includes a lot of different proteins circulating in the plasma. Initially, these proteins are not active. But in case of contact with pathogen-associated molecular patterns, some of them become activated and then activate other proteins that belong to complement system in a cascade-like manner. Activation of the complement system proteins results in the formation of the structure known as membrane attacking complex. This membrane attacking complex makes holes in the membranes of the pathogens, leading to its destruction. Additionally, some byproducts of complement activation attract immune cells, guiding their migration towards the pathogen. And also activated proteins of the complement system that are attached to the surface of pathogen help immune cells to detect their targets. Now let's speak about adaptive immune response. Generally speaking, it has two parts: cellular components and defensive proteins acting outside of the cells. Cellular components of the adaptive immune response are represented by T lymphocytes and B lymphocytes, while defensive proteins are represented by so-called antibodies that are produced by activated form of B lymphocytes that are called plasma cells. It is important to note that adaptive immune response and innate immune response are not isolated but actively cooperate. For example, dendritic cells that belongs to innate immune response play an important role in the activation of T lymphocytes, while antibodies that belongs to adaptive immune response help to activate phagocytic cells that considered to be a part of innate immune response. Now, let's look at how T lymphocytes work. Here you see a schematic illustration of the antigen, a structure that could be specifically recognized by the T lymphocyte with the help of T cell receptor, or TCR. This TCR is located on the surface of T lymphocytes. Important point about TCR is that it cannot recognize antigen without additional help. To make antigen recognition possible for TCR, antigen should be presented to T lymphocyte with the help of so-called MHC protein. It is called so because it is related to the genes of the major histocompatibility complex. So now, when antigen is presented with the help of MHC protein, T lymphocytes may recognize it with the help of T cell receptor. And now comes the question, what the cell will do next? There may be possible several different scenarios. In case if partner cell is infected with the virus and antigen that is presented to TCR is related to genetic information of the virus, then activated T lymphocyte may kill the infected cell. A similar action may happen in case if partner cell undergoes oncological transformation and presents a mutated antigen that normally should not be present on the surface of the healthy cells. Such T lymphocytes programmed to kill the cells upon activation are called killers, or CD8 positive lymphocytes. This is because they have a special structure called CD8 on their surface, and interaction of this structure with MHC molecule is important for activation of killer lymphocytes. Now let's consider another scenario and imagine that partner cell for the T lymphocyte is a healthy antigen-presenting cell that presents antigen related to the pathogen. Since this cell is healthy, it should not be killed. So, what T lymphocyte should do when it detects antigen related to the pathogen on the surface of healthy antigen-presenting cell? Instead of killing friendly cell, it may release biologically active compounds typically called cytokines, and these cytokines will stimulate other cells to fight with the pathogen. Such T lymphocytes that upon activation stimulate activity of other immune cell types called helper cells or CD4 positive lymphocytes. This is because they have a special structure called CD4 on their surface, and its interaction with MHC molecule is important for activation of helper lymphocytes. However, this is not yet the end of the story about the activation of helper lymphocytes. This cell additionally carry a structure on their surface that is called CD20. To support helper cell activation, this CD20 structure should interact with the coactivator structure on the surface of the antigen-presenting cell. If this coactivator is expressed on the surface of antigen-presenting cells, helper lymphocytes will be activated in response to antigen recognition. But in case of the absence of coactivator on the surface of antigen-presenting cell, helper lymphocyte will not be activated. Why it is important? Let's imagine that the presented antigen is related not to the pathogen, but to normal healthy cell of the body, so-called self-antigen. In this case, production of stimulatory cytokines by helper lymphocytes is not reasonable and should be prevented even in case if occasionally self-antigen will match with TCR, T cell receptor. And this is achieved by the absence on the surface of antigen-presenting cell that show self-antigen co-stimulatory molecule for CD20. In contrast to this, when antigen-presenting cell process antigens that are related to pathogen, co-stimulatory molecule for CD20 will be expressed on its surface, and this will make possible helper lymphocyte activation. And of course, this in turn will lead to stimulation of other immune cells that are involved in pathogen clearance. Therefore, CD20 on the surface of helper lymphocytes, together with inducible expression of co-activator molecule on the surface of antigen-presenting cell, contribute to a safety mechanism that prevents helper lymphocyte activation in response to self-antigen. Also, there is another special type of lymphocytes that produce anti-inflammatory cytokines suppressing the activity of immune cells. This is very important because uncontrolled activity of immune cells may lead to significant collateral damage to the tissues. Such cells that instead of activation of immune cells will suppress their activity, called regulatory T cells, or in short Tregs. Other cells that are involved in adaptive immune response are B lymphocytes. These cells can recognize a wide range of pathogens with the help of B cell receptor, which in contrast to the TCR does not require support from MHC protein. Upon activation, B cells differentiate into plasma cells, which produce a lot of antibodies working as soluble receptors able to recognize and bind to antigens. Such binding of antibodies labels pathogens carrying these antigens for elimination. There are several different classes of antibodies, particularly IgM, IgG, IgA, IgE, and IgD. IgM, IgG, and IgA together compose more than 95% of circulating antibodies. I hope that this overview of immune system was clear and interesting for you and will help you to study further more detailed knowledge of immunology.